So I want to end with telling you a little bit, maybe for the next 10 minutes or so, since today's topic was translation, so I guess I'm going to have to stop using that word or Ray's going to get mad at me. But future medicine, how do you do this? What are some of the issues? So I thought you might be interested in actually seeing, what happens, some of the things behind the scenes about when you take fundamental science and have to actually convince the FDA to be able to allow you to treat a patient. So you start off, and let's say you get some nice results in an animal model like we did here at Caltech. What's the three most important things when moving this into clinical trials? Money? That's the first one. What's number two? Money? What's number three? Time. But you're right, because time's money, right? So I really feel that this is the issue. It's really hard for us on campus. We go and we get our grants and so forth, but this is a whole nother scale of finance that's necessary to do this kind of work. So if you have a blank checkbook, which no one ever has, this process the FDA has is very straightforward. The problem is it's not straightforward when you don't have a blank checkbook, okay? So the normal situation is you try to do the most intelligent decisions you can with limited funds to try to convince the people at the FDA you're not going to do any harm, okay? So what do you have to do? Well, if you treat a patient in the United States without FDA approval, you get thrown in jail, okay? Plain and simple. So you have to go to the FDA, you have to get approval to do so, and you have to fill out this application called an investigational new drug application. And so you might have heard this called an IND for short, okay? This contains three broad areas of information. The first is your studies on animals. What did you learn? How did you make this thing, and can you convince the FDA that you can do it? These are very safe, you worry about impurities, et cetera. And then what are you going to do to patients and why? Who are going to be the physicians who are going to treat these patients and so forth? Now, how many of you remember, doing your PhD thesis? It was like, oh man, what a pain. Here's an IND. Okay? A PhD thesis is nothing compared to one of these. I've done two of them now, and we're in the midst of doing a third one. These are thousands and thousands of pages of information. Okay? So these are not fun, okay, to put together, but there's a prescribed process to do it. But they're very, very large documents with huge amounts of information. So what's different about cancer is, in other areas of drugs, you start off with healthy volunteers in the trial. In cancer, you don't do that. And it's traditionally because these cancer drugs have been so toxic that you don't want to put them in a healthy person. So these phase 1 trials turn out to be patients where all the other standard therapies have failed to help them with their cancer. So most patients you get here are in really, really bad shape. Okay, so you're starting off with absolutely the worst patient population you can see. So you have to determine what you're going to do in here is determine the safety here, and then decide what's the right dose so that you can continue on with your trials. So the key here issue is when you negotiate, and it is a negotiation with the FDA, is you have to develop a scientific rationale for how you're going to dose the patient and what is that first starting dose. You literally spend millions of dollars getting that one number, okay, that you agree with the FDA. So you start off and do these animal experiments, and you have to do a minimum of 2 species. One can be a rodent, but the other has to be a non-rodent. They're very expensive. So for example, in the RNAi case, when we did the non-human primate study, that study alone was $1 million just for the animal work. And I was so nervous about this because it was so new that we, instead of doing 2 species, we did 4. Okay? We wanted to be absolutely sure of the safety of this. So what you find from the animal work is you determine what's called the no adverse event level. So you study all these species and you find out where you have no event at all, nothing in any of the animal. And then you typically go at some fraction below that is where you start in the human, okay, to try to be as safe as possible. Now, in a cancer trial, why is that important? Because these are real cancer patients. If you go too low, then your hope of doing anything with them is minimal, okay? So you have to think ethically in these trials. And it also costs you a lot more money. But you can't go too high because you don't want to hurt somebody. So you have to be careful about what this window is. And this is an important point that people always forget. We determine the toxicity of these in healthy animals. That's not the same as a sick human. Because these patients in these cancer trials are very, like one lady stuck out, I remember in the first trial, she had had 11 different therapies for breast cancer before we started into this trial. I mean, she had no bone marrow left. She'd walk into the physician's office and start throwing up before you even started talking to her, so she was so used to it. So you're dealing with very sick people in these very first trials. And the key issue here was, this was the first movement into this area of science. How do we translate information from an animal to a human? What's the scaling factor? No one knew. So the FDA said, well, if you don't know, we're going to tell you what you're going to do. You're going to do what we do for drugs, which is the scale on body surface area. And I said, well, what if that's not right? He said, well, you don't have any better data, that's what you're going to do. So guess what? They were right. But we didn't know that. Now we do. So if you look, what this AUC is, so this is the circulation in the blood, as I said. If you measure the amount under the curve of that, so that's the AUC, area under the curve, that's like the exposure that you have of your drug to the animal. So if you look, here's rat, here's dog, and now here's human. If we scale these on body surface area, it scales very nicely. So the FDA was lucky. It was the right scaling for these nanoparticles, but no one knew at the time. So now we know this is the way to do this in the future. The other thing is, you're not going to treat a cancer patient with one dose. You're going to treat them for multiple doses. And so you do animal experiments where you treat for doses for several months. And it showed that it's hard to see here because they're right on top of each other, but this is dosing for like 3 different months in these 2 animal species that showed there's no difference in multi-dosing. And it was supported, and when we went into humans, the dosing here, it's right on top of each other, is at 1 month and at month 6. So there's no change in the dosing as we do this for many months. And here are the hard data, which again, it was nice that the animal data were supporting what we were seeing in humans. Now the key is that we want to make this material, or whatever your therapeutic is, at high quality and purity, and it has to be sterile and so forth. And you always learn a lot as you start to scale this up. Now here at the university, probably when you were here, everyone always told you want to make everything absolutely pure, right? 100%. If you can't do that when you go large scale, it's impossible. And if you make it too pure the first time, now you're in trouble, because if you go and do your animal experiments at a certain purity, your human material has to be even purer to support that your toxicity of your animals will move to humans. So I always tell people you don't want to be as good as you were when you were a student. You want to be less pure. And I said this one time, and there was somebody in the audience from the FDA, and they got all mad at me, and they said, what are you doing telling people not to make it pure? I didn't mean to say that. You're still making it 95+% pure, but you actually want to see what some of these impurities are and see if there are toxicity issues in the animals from those impurities. So you want to think about all of these things ahead of time. So it can be really important, and this is the key. You know, you can do pretty much what you want as long as you can convince the FDA that you have reproducibility. And so, for example, in this process, we developed a process to make it into the vial. And you know, you do this at larger and larger scale all the time. You use sophisticated instruments like we have here at Caltech. This cryo-TEM is, you know, a multi-million-dollar instrument to be able to see the particles and measure the particle sizes. Obviously, as a company, you're not going to be doing this, and you can't do it reproducibly, but it teaches you that then you use other techniques like light scattering, which can be done reproducibly on all of the things you're doing commercially. You have to worry about your drug. It has to sit on the shelf. You'd like to have 2 years stability, possibly 4. You also have to make it fall apart for the FDA, because they want to know that you can actually detect when it falls apart and what it falls apart to. So what's interesting is that this is a lot of analytical chemistry, and most chemistry departments in the United States don't teach analytical chemistry anymore. So it's hard to find people who have backgrounds in this area, but this is all analytical chemistry. You have to worry about stability in the clinic, and this is a big one, is when you put this in a bag and infuse it in the patient, remember, these are all plastic, and lots of things stick to plastic. And so there are examples where someone put a drug in the bag, as it was going down the line, it stuck to the line and never got to the patient. So today, you have to go and get all of these infusion sets and show that what's going on even before you're allowed to treat the first patient, so that that doesn't happen again. So the question is, when you're making this, you say, I'm going to have this set of properties, and then I'm going to have error bounds on that property. How do you know what those error bounds are when you've never gone into a human yet? What do you do? It's you know, the old chicken and egg problem, right? So what I decided to do was to say, okay, in our case, what was important was the molecular weight of the polymer and the drug content. So what if we just do a simple 2x2? High molecular weight, high drug, high molecular weight, low drug, low molecular weight, low drug, high molecular weight, low drug. So we make all of these materials and we study these in animals, and we were able to show that within this parameter space, the properties, the function in the animal was the same. So we used that information to convince the FDA that within this parameter space, we would expect it to behave the same way in a human. Okay? So that, if you have any other ideas, I'd like to hear it, because this is a real problem. How do you think about doing this? So then, these are the commercial batches that were made. These are the sizes of the particles by the dynamic light scattering. This is by the cryo-TEM, just to confirm we did it right. And so we start to make these, and then when you get your first experience in humans, now the good news is for us that approach worked when we kept those parameters within that particular range. What we were able to see was very nice reproducibility. So I know the numbers don't mean anything, don't worry about them, but so here were 24 patients that received this lot, and here were 18 patients that received this lot. The data were very, very consistent from these patients. And so you build up now your own database from humans, where you ultimately, by the time you get approval, you have it worked out in humans. So phase one, just to summarize, phase one is that you get your initial look, you get safety. Phase two, you continue to improve making your drug. You're doing more and more patients. You start to close in on how to manufacture it, and then by phase three, it has to be fixed. You can't change it once it's that point. So I want to end with just 2 or 3 slides saying, okay, I showed you a lot of data about what can be done in cancer. Now at Caltech, we're trying to figure out how things other than cancer can be used. So here's what your kidney looks like. And those 10 nanometer holes are out actually in this part of your kidney, which are called tubules. But you also have this area in your kidney. And there's a lot of function in this area that occurs. There are cells in this area called mesangial cells. And unfortunately, these are the cells that start to go wrong when you have diabetes and so forth. So when people start to have renal failure, it's this part of the problem for diabetics. So when these drugs go through your blood vessels and then they go out these 10 nanometer holes through the tubules, you can't get and do therapy to these cells. But through fundamental studies here at Caltech, we were able to show that now we can design particles of a different size. So it's no longer this 30, 40 nanometers. This size to get into these cells in that area of your kidney should be around 80 nanometers. Now don't ask me why they're different. Okay, I have no idea why nature's done this. But it's clear we can hit every one of these if we tune the properties of the nanoparticles now to hit this cell. And this is just to show you. So if we put, again, a fluorescent probe, here's a drug molecule. So here's the area where those mesangial cells are. You see, it doesn't go there at all. It goes out those tubules. When we now do it with a nanoparticle, which here are a picture of the nanoparticles, look at this. The distribution of kidney is entirely changed. It's completely from going out the tubules to going into those cells. And so we're working with collaborators other places to see if we can now bring therapy to those cells that would be effective against people who have diabetes. And finally, I want to tell you about work that we're currently doing with several MD, PhD students here at Caltech. You'll hear more about the brain today, but you have this barrier called the blood-brain barrier that separates your blood from the cells inside your brain. So this is the problem with getting therapy into your brain. There's very, very few molecules that go across that blood-brain barrier. And we've now, for the first time just this year, we've published our first results where we're learning the rules. What are the design rules to build particles that can actually go across the blood-brain barrier? So for example, here's a section of a mouse brain where the particles are the dark color that are in, you can see these are in your vessels, and they're not getting out into the brain. Where when we do it right, now you can see all these particles out into the brain. Okay, so where this is some of the current work by a couple of MD, PhD students, where now we'll be going with people who understand things like Parkinson's and Alzheimer's and see if we can now develop ways to make therapy go to the brain. So with that, I'm going to end. I get to work with great people here at Caltech and the companies. I'll just leave this up here for you can read it during questions and answers. For me, this has been the most amazing thing, is that actually the physicians have let me in the treatment rooms to talk with the patients. You know, they're always very helpful. And of course, I have to give you my conflict of interest statement, which is there at the end. And so I'll be happy to answer what questions I can for you for the next few minutes.
Thank you, Mark. Patsy and I will be taking questions from alternative sides of the room, and please wait for the mic so we can record your questions for alumni who will watch this session on iTunes U. Our first question.
That was really fascinating. I'm so glad that you brought up the blood-brain barrier at the end. So that was going to be one of my questions about passing through that. And I was also thinking about, you mentioned certain types of cancer, but are these equally effective in various types of cancer, and including brain cancer?
So what people do, even though you generally classify cancers by organ, you know, like it's breast cancer, that's really not the way today. People are trying to get away from that classification and actually say, what is the gene that's driving that cancer? So you can have a particular gene that's driving breast cancer, that's the same gene that's driving prostate cancer. So today, when you're trying to develop new cancer therapeutics, you do it as what is the gene or target you're going after. So that's why you might see one of these go for multiple different types of cancers, but it's actually going at the same target. So as long as you're attacking a target, it looks pretty good. Our phase 1 trial that we did was one of the very few in the country that allowed brain cancer patients into the trial. Usually a phase 1 trial excludes brain cancer patients. It works if the disease is metastatic to the brain, because what happens if you have a metastatic cancer in your brain, that blood-brain barrier is breached and it's leaky, so you can come in from the blood system. When it's primary, it's not going to work. So that's what we're working on now, is how do you get these particles across what I would call a pristine blood-brain barrier. Okay?
That was one of the most fascinating talks I've heard in several years of alumni college, so thank you very much.
You're welcome.
Though I was not surprised by the extent to which the FDA process and rules drive what you're doing to get it to humans. But the FDA, Caltech, after all, has a global, if not galactic scope. The FDA is not the only regulatory agency that approves such things. Have you worked with or considered working with the ones in Europe and Asia? And their process may be better, may be worse, but it's certainly different.
You're absolutely right. And each of these agencies have different subtleties. They haven't harmonized the whole world yet. So like the European agency is different than the FDA, and the Japanese agency. And the newest one that's come on now, people are going, you're going to see more trials come out of Australia. And it's not because of the agency, it's because the government now gives you financial incentives to do those trials in Australia. So watch, you're going to see a lot of trials come out of Australia. The 160 patient lung cancer trial that I showed you the safety data from, that was all done in Europe. So we're approaching both pathways. The European agency has some advantages, it also has some disadvantages. So it's really amazing how it really gets down to when you get close to the final goal that you sit down and you actually have to talk with them and see who's going to be the better of the two.
Okay, Mark, I'm a dictator of the United States, and I have now just given you a blank sheet of paper. How would you do the FDA differently in this process?
Yeah. No, it's a great question, and people are struggling with this. I think those people have a very difficult job because every time that they try to help out, well, I should maybe not use the word help, every time that some of the processes go astray, and then we have a problem like the Vioxx problem, then they get killed, right? People in Washington just go crazy. So I think at this point in time, the process itself is probably okay. What it is they don't have enough money to be able to do their own experiments to when they have questions, they can't go answer them. So what they have to do is fall back to positions that they've done in the past that with new therapies, those are probably not good fallback positions. If you want a hard, concrete example of that, I can give it to you. But I really think that they're underfunded to the point where they're not able to make decisions as well as they could if they had money to go and answer certain questions in their own mind. Because again, they have to be skeptical of all the rest of us, okay? Because of what's happened in the past, they're skeptical of companies. So they need to independently verify certain things. So like when I did, that Senate panel, that was a question that one of the senators asked me. He said, well, what should I do? Tell me what's the number one thing I should do. And I said, give more money to the FDA. Okay? And that's what everybody always says, right? They want more money. But in their case, I think they really are underfunded to be able, because the science is moving so fast that they can't keep up with it right now.
Just like, well, I think that patent law is not taught to engineers, even at Caltech. And in this case, you've been through the clinical trials, and so you know a lot more now about how to structure your work because you know what to prepare for, having been through that process. Do you teach any of the Caltech students what you've been through? And why not, if not?
There have been several occasions to have interactions like we're having today. There hasn't been an occasion to basically have a course or have a more formal setting where you could go through a whole series of things. So, because there is the science part of it, it's the question that was raised about the FDA. There's lots of things with the FDA you'd like to be able to influence the FDA. There's also all the IP. This is a tough area of IP because now many countries in the world want to get into this game. So if you start off and you patent only in the United States, you're screwed. Okay? Because other countries will just take this stuff and it's all gone. So a place like Caltech, actually we're doing, I think, a pretty good job here in the Patents and Licensing Office where we go and start and we do worldwide patenting in addition to US. In this drug area, if we didn't do that, none of these things would ever have any potential for commercialization because virtually every country in the world is putting up facilities. You can see, India, Singapore, Israel, Australia, China. Everyone questions whether you should even pay for China because they tend to go around everything you do anyway. So we've had some of these, like I said, we've had some of these kind of group sessions, but there's no formal course here or anything.
Great job, Mark. I think that we started this morning by talking a little bit about, or listening to thoughts about how public policy wants to translate or reap the rewards of all the investment that they've made in medicine. And I think that your example of talking to the Senate really epitomizes how the enormous gap between the public, the politicians, and their understanding of the process of bringing medicine to people and what actually has to be done in the detailed work to actually have that accomplished, whether it's statistics, whether it's funding, whether it's timelines, et cetera. And I'm a firm believer that the FDA is good. It's a good process check. But also in our generation, we've seen a mentality change from the man on the moon will do everything to get this goal accomplished to the VC mentality, which will sprinkle 1,000 seeds in hopes that one gets through phase 1 and that'll weed it out. And then maybe someone will get through phase 2 and maybe we'll get to phase 3. How much do you think that mentality, man on the moon versus VC sprinkle the seeds, affects a process like this in enabling you or other researchers to really bring taxpayer investment to cures?
Yeah. Well, the reason I'm hesitating is because I've been very outspoken about this, and I don't want to get myself into trouble today.
It's all recorded.
Yeah, that's right. So I actually think the whole paradigm, the way the pharmaceutical system is doing it right now, is got a problem. So here's why. Those of you that are in other industries, think about your industry. The success rate in the pharmaceutical company right now, on average, is about 1 in 30. So think about whatever your industry is. If you had a success rate like that, could you survive? It's pretty bad, right? Okay. So I think that what happens is then when a therapy gets approved, you have to pay for 29 failures. So these costs are huge at that point. The NIH is great. It's great for discovery. I don't think it's going to be great for translation. So I actually think this $1 billion that they've now decided to put into translational medicine is probably not a good idea. Okay? Those are good people that do good fundamental work. They're not great at making drugs. Okay? That's not their objective. Companies make drugs. They do fundamental science. But I do think that we have a problem. And there's been some recent discussions over the last couple of years about trying to create new entities that would take big funds and develop new paradigms to attack diseases. So you create a mega fund for cancer, you create a mega fund for Alzheimer's disease, and then what you have is different ways to finance what you're doing. Ultimately, you bring the pharmaceutical companies in because they have the way to end off the phase 3 trials and to do the marketing. So I'm not really answering your question other than to say, yeah, I agree. The pharmaceutical system really needs to rethink what they're doing because it's kind of broken right now. But there are all these issues because of this process. So let me tell you the one that's really difficult right now is if you talk to people who make imaging agents, so there's all these great new imagers and imaging agents, you can't go through trials and pay for that kind of money, get a new imaging agent approved, and then get your money back on what it costs you to go through those trials. So all these companies are saying there's all this great new technology coming from these universities, but we can never commercialize it because we can never recover our money. What I'm trying to convince people is that we should go to the insurance agencies, the HMOs, and say, look, if you're going to pay for a $100,000 therapy, don't you think we ought to image this patient and make sure at least we have a chance of that therapy working in the patient? That if we could somehow get the insurance agencies to drive saying, I'm not going to reimburse you for a $100,000, $200,000 therapy unless you've done the imaging to show me that at least we have a chance in that patient, to be able to help do this. So there's all of these kinds of things now that are coming under discussion, but I do agree with you. The system is kind of out of whack at this point when you're talking about the way that's going on. You know, going back to the question of the FDA, I still think the process they're trying to do at the FDA is right because you want to be as safe as possible before you let it out in the public.
Hi, thanks for a great talk. Very exciting. I work for an imaging company, and I actually had an imaging question before you brought it up.
Okay. So do you agree with what I said?
I do, actually,
Okay,
very much
thank you.
so. But my question was, you use a lot of imaging tools, SIMS, PET, a lot of digital pathology with fluorescence markers. What tools should we be giving researchers like you? If you could design a new tool to help you, what would it be?
Well, I think that the biggest impact that's had on research for many different reasons are these whole body imaging tools like PET, like SPECT. I have mixed feelings about MRI. It's getting better, okay? And the reason is that in the old days, we used to do what I used to call slice them and dice them. So if you were going to go and understand a study, you had to do many, many animals, sacrifice the animals at all these points, take all the organs, do all this work. Now with an imager, we use far less animals. It's makes so much more sense from an ethical point of view to get even more information because we can see what's going on in the whole animal in the same animal. So the idea of doing whole body imaging has revolutionized the way that you can do science and translational medicine. I don't know what it would be, but if you could get a imager that could give you the resolution, this standard thing. If you could get the resolution of an MRI with the sensitivity of PET, you could solve so many problems for us right now. So we like PET because we like the sensitivity. Okay, most of these, when we do MRI, we're on the edge of seeing what's going on. I like the PET because of the high sensitivity so that I can't miss something. You can't go hide somewhere in the body without being able to see it from the PET. So, but that's the one thing is PET doesn't have the resolution. And so when we go in the patients, we've been doing PET CTs on patients just to get an experience with that. The problem is it's really expensive. When we did PET CTs in the trial, it doubled the cost of the trial just to bring the PET part into it. So anything that could be done to make that cost more reasonable, we could bring into more trials earlier on.
Mark, I know nothing about biology or medicine, but I am an engineer trained at Caltech. And I agree with your statement that money, money, money of getting the most bang for the buck is extremely important. Mm-hmm. And they taught me in engineering that if you think of the right format of the differential equation, you could generate your chart with only 3 data points. And I noticed a lot of your charts were very steep fronted on the forward end. And you might try replotting it on log scale, and it may linearize it, and it may give you more insight into what's going on.
Okay.
Thank you for the fascinating talk. A few days ago, I read in the Wall Street Journal saying that FDA is considering or maybe actually using the fast-track method for the target treatment because often the success is so spectacular, you don't need to go to 10,000 patients. Is that true?
Yes. So there are accelerated pathways through the FDA if you're going after something that has a very high unmet need. And and they're trying to get more of those in place. Let's go back to the question of VCs and funding and how we have to do things now, because it's tied very much into this. So here's what happens often, not all the time, but often. So you get this big investment into your company, and so you say, I'd really like to go after that. For accelerated approval, it's a high met need, and they go, it's not going to bring back a big enough rate of return even if you're successful there, so we don't want you to go there. You make sure your therapy goes over here because there are many more patients, say, in breast or prostate or so forth. So there's a lot of really good science that if there are a way to fund it to go after these high unmet needs that are usually much smaller patient populations, the investment community often, because the risk goes up, as you might expect, those are even higher risk, they don't want to go there. So they tend to swing you in other directions when they're putting in that kind of money, they basically tell you what to do. But these new pathways are really good. So if you can make it go into one of those new pathways, it's going to be very helpful.
I'm a physical therapist.
I'm
over here. I became a physical therapist after studying biology here at Caltech, and I spent years at USC, including Norris Medical Center. One thing physical therapy and occupational therapists do is help improve patients' quality of life. And with cancer patients, we get them at the very end. Doctors are all excited that they're successful, they've treated the cancer, and these patients are totally debilitated, can't get out of bed. I just wanted to commend you for your work, for looking first at their quality of life and trying to bring everything full circle to help the patient end, and not just curing the treatment, but curing the patient as a whole. So on behalf of all the patients and healthcare professionals who struggle day to day helping these patients, thank you for looking at the big picture and going all the way back to your molecular research. And I hope you inspire a lot of people in the future to do research like you have, to go full circle and look at both the patient end and the science end of it. So thank you.
Thank you. No, it's very personal for me that the picture of the person that I used as the example as the problem was actually my wife. So, you know, I lived through this for quite a bit myself. But I think it is, and the FDA now, you actually do as you probably know, quality of life surveys in later stage trials, so that's an important piece. So, as you might tell, I like to tell stories, so let me tell you one about. So, in these trials, we are seeing high quality of life. And so patients are taking their therapy and going on with their life, but it also creates interesting scenarios when you're doing a clinical trial. So we had a particular patient who was feeling so well for the first time, went out into the desert, was riding, you know, these RTVs and fell off and broke both wrists. Okay? So he was so happy to feel good. So he comes back to the hospital, and I'm like, well, okay, he was just being an idiot. No, it's a trial. So the first question was, this is a new drug, is it affecting his brain? So after a lot of money was spent on neurological tests on this individual to make sure that it was just he was having fun and had an accident and it wasn't a neurological problem. So as I always tell my students, you know, these are not lab rats. These are humans who go on with their life when you're doing these trials. So that's what we'd much rather have that than the other case, okay?
That was a great talk, and I have seen the video, and I encourage other people to see it, and I'm looking forward to showing it to my daughter in Chinese so that she can practice her Mandarin. Shameless plug, I work at USC now, and I work with engineers and doctors, and anytime you have a student who wants to sort of cross the engineering medicine barrier, please, let's talk. I would caution against future medicine, the term, because people with family members who need help now are often, you know, discouraged by terms like that.
Yep, no, you're right, you're right.
And I love your idea about how do we change the paradigm. I mean, this is the key paradigm shift, because science has progressed so quickly. How do we change the paradigm for actually doing, you know, better translational medicine? And I think, you know, there are outfits out there like Faster Cures that are, you know, the Milken Foundation. But I think, I really think that we should step back and look at those processes and get a real paradigm shift there too. So I'm interested in some of your thoughts along those lines too.
Yeah, no, it's a good point. You should tell Ray when he comes back too. We do, the really nice thing about Caltech now is we do have MD, PhD programs with both USC and UCLA. The two people who are doing the blood-brain barrier project are both MD, PhD students from USC. So if you'd like to meet them, come over and chat with us, okay? So we're already doing things with USC.
I was thinking along the same lines, that future medicine is way out there in the future. There's no, and the translational medicine, the way I understand it, is speed up the time between benchtop to bedside. So isn't there something kind of fundamentally at a friction here where you show a slide that this takes a long time and gets away from sort of the fundamental science that's done at Caltech to let's get this out into the marketplace? That's sort of not what Caltech is, and I'm sort of wondering, however, because there's such a need, are there any plans for an institute for translational medicine? I know there's one at the University of Pennsylvania.
Yeah, so first of all, most people don't associate speed with the word translational medicine. Translational medicine is usually how do you take it from a lab into patients. That's traditionally what that term's been used for. There's a lot of discussion on Caltech's campus about formalizing this. The only thing that's happened so far is that David Baltimore was able to convince Eli Broad to give a $7 million seed fund to Caltech to try to help stimulate between Caltech, UCLA, and USC these interactions towards translational medicine. It's very limited, to be honest with you. Most of it's still, here's some great fundamental work that's been done at Caltech. How in the world do I take the next step, and how in the world do I get the funding to be able to take the next step? There's so many, as Ray said, you know, when you really sit and listen to the colleagues across campus, there's so many cool things that are coming, but they're all those two factors. It's not only the money, but they don't know what to do, how to translate it. So to formalize it would be good. To be blunt, there's been a lot of talk about it on this campus, but, you know, very little action. So, you know, I think the administration needs to put, you know, their money where their mouth has been, to be blunt, because I think it would be interesting, but we don't have anything here.
For one or two more questions.
Thank you, Professor Davis, for that talk. I actually just graduated last year from here, but going back to a couple questions earlier, you said that your current research is now working on crossing the blood-brain barrier with these nanoparticles, but of course you yourself admit that it's not perfectly localized. You always hit the other healthy cells around your tumor cells. So how would you convince the FDA that these nanoparticles that are entering healthy brain cells aren't going to affect people's state of mind, you know, state of health, well-being, and just their own, you know, psychosomatic processes?
Very carefully. That's how we have to do it. So, you know, it's a really hard problem, as you might expect. So this is why we go back to fundamental science. There's lots of claims in the literature of people crossing the blood-brain barrier. When we've gone back and tried to look at those data, a lot of times what they do is actually destroy the blood-brain barrier, and that's how these things get into the brain. That's not going to work if you were ever to get to a human, right? So we're going to have to be very, very careful here. And so it's just going to be a lot of, you know, very sophisticated animal work prior to being able to go into humans the first time. You know, I'll admit, it's, you know, the cancer problem is a hard one. I think the blood-brain barrier problem, getting in and doing some things neurologically, is even going to be harder. But that's our style. Like I said, I'd rather work on those really hard problems than, you know, some other ones. But I do agree with you that that problem, you're going to, you know, when I see people talk about other things that they say are crossing blood-brain barriers in animals, man, I would, if I were a physician, I wouldn't go anywhere near it trying to bring it to a patient. So we're going to have to do an enormous amount of work at the animal level to convince a good clinician that it's worthwhile to go into a patient.
Question up here. Early on in your talk, when you were talking about the sort of ideal properties of a drug in addition to the effectiveness, you said that you wanted something that was low cost, and you also talked about the cost of cancer therapeutics right now. And I would assume you're not going to have any influence on how much Cerulean is going to charge for your drug once it's approved, if it should get to that point. But I was wondering what your thoughts were, because it seemed like maybe you had some thoughts about where you wanted to go with that.
Yeah, I'm sensitive to the cost, because I think in the future you're going to, it's the same thing that happened with HIV, right? HIV, they had a real problem doing anything in patients when you used an individual drug. As soon as you got to attacking at 3 different positions, you started to have a real effect on HIV, right? So it's the same thing with cancer. You're not going to have a real effect on a patient by only attacking one position. You're going to have to attack multiple targets simultaneously. And so that means you're going to have multiple therapeutics. If every one of these things is $200,000 of therapy, now in principle you could get up to $500,000 therapy. That's just not going to work. So we are sensitive about how we can bring multiplexed therapy to patients for acceptable cost. The problem is today the way therapeutics are priced, they're priced to whatever they can get. So it isn't correlated to what it costs to manufacture. It's correlated to what they can get for that treatment. So when we ballpark out the cost of that first nanoparticle to make, it's actually very, very small. The question is just what you said, what will they ultimately sell it for? I know I'll cringe when I see it, okay? Compared to knowing what it actually costs the manufacturer is going to be quite low. So, you know, there just has to be more discussion. Like these discussions, I don't know whether you've seen what's happening in England these days as far as the British system saying no to certain very high-cost therapeutics, saying we're not going to buy into these high-cost therapeutics. So there has to be some of these discussions to help bring them down. But again, remember that you're paying for 29 failures with that too. So that's the other piece of the problem that we have to worry about.
Okay. I think we've reached the end of our time. Thank you so much.